Hi, my name is Rachel Diaz, and I'm excited to talk about a potential way to diagnose biofilms in the future. I wanted to start off by asking you all to imagine that instead of your mask blocking out particles of infection, what if it was like a sticky web that allowed pathogenic particles to attach to it and become stronger and more infectious? Now imagine that you had no idea that you had this defective mask and you weren't getting any better from an infection for over 6 weeks. This is exactly how biofilms are in nature, and it is a huge problem in microbiology and infectious disease. So, this will be an overview of what I will be going through today about my research. And going off of the analogy that I just mentioned about masks and biofilms, I first wanted to talk a little bit more about what biofilms are and the problems that they cause. A biofilm is a group of bacterial cells that come together under stress and excrete what are called extracellular polymeric substances, also known as EPS or the extracellular matrix. The EPS acts as a barrier to protect the bacteria, and it provides a network of communication between them. Although biofilms are beneficial to bacteria, they can be problematic in the human body. One problem is that biofilm infections are a lot harder to kill than non-biofilm infections. They usually require more aggressive treatment, and because the EPS allows the bacteria to have better communication, antibiotic resistance can also happen a lot easier. The longer an infection persists, like in chronic wounds, for example, the higher chance of developing a biofilm. It is seen that 60% and up of chronic wounds are susceptible to developing a biofilm. Visual cues are not always present, and it takes a long time to diagnose a biofilm in the lab. One group particularly susceptible to infection are people with diabetes. According to the CDC, diabetes is the seventh leading cause of death in the United States. Because of their suppressed immune systems and poor circulation, people with diabetes have a higher chance of developing a chronic wound than the average person. This is a huge problem because the CDC reported about 10% of the US population in 2020 to have diabetes, and another 35% of the adult US population were reported to being pre diabetic. That is almost half of the US population at risk of developing a chronic wound at some point in their lives, and thus they are at risk of developing a biofilm infection. Moving on to how this problem of biofilms can be solved, I want to talk about the company Moleculite and how their device could potentially be the answer to diagnosing biofilms in real time. Moleculite is a company based in Canada but has FDA approval in the United States in order to sell wound imaging devices. Their wound device, the Moleculite IX imaging device, is used for the diagnostics of bacterial infections. The camera has also been shown to work regardless of if the bacteria is encased in a biofilm extracellular matrix or not. The left picture on this slide is the basics of how the camera works, and the right picture shows the different angles of the imaging device, so you can roughly imagine the size of the camera and the different characteristics that it has. The camera consists of an iPod encased in the actual Moleculite technology that is built around the iPod. When the lights are off, the Moleculite IX imaging device is able to shine a violet fluorescent light that is around 405 nanometers, and three different outcomes can happen when the light hits the wound. The first option is that the wound will fluoresce a green color, meaning that there is no bacterial infection present, or at least not one that produces porphyrins. The next option is that the wound will fluoresce red, meaning that there is a bacterial infection and a swab needs to be taken and a course of antibiotic treatment might be needed. And the last option happens in specific bacteria like Pseudomonas aeruginosa. Pseudomonas aeruginosa produces a different kind of porphyrin that is responsible for a cyan light to fluoresce instead of the standard red fluorescence. But in this case, we will only focus on the standard red fluorescence. Under normal circumstances, this device was built around a natural biochemical process in our body called heme synthesis. Bacteria like to exploit this in order to provide themselves with more nutrients, mainly iron. The highlighted parts are the most important parts of this cycle in regards to the molecular camera. Aminolevulinic acid, also known as ALA, is how we can induce lab-grown biofilm bacteria to produce porphyrin. After going through a lot of steps, protoporphyrin IX is made, which is what causes the bacteria to emit the red light that we see under the camera. This next slide shows the visual chemical structure of porphyrin on the left, which usually complexes with the iron 2 plus to form heme, and on the right is the structure of ALA that we add to the bacteria for them to produce the porphyrins that they need in order to glow red under the camera. Next, I will show a quick visual example of how the camera works. I did a streak plate of Staphylococcus aureus, which is a common wound pathogen, instead of providing actual pictures of wounds because they can get a little nasty with tissue death and all of that. On the left, the staph is plated on blood agar, which has 5% sheep blood, but no components in it to produce porphyrin. And on the right is a porphyrin test agar with ALA in it to induce porphyrin-producing bacteria to produce porphyrins. When we turn off the lights, we can see that only the plate on the right glows red because only the porphyrin test agar has the nutrients necessary for the bacteria to produce porphyrins. This next picture is a side view of the fluorescence, and I wanted to show it to show that this can be seen not just in the device picture, but if you were to take your own violet 405 nanometer light and shine it on the bacteria, they will glow red. The camera is used for the convenience of collecting pictures as proof, and it can even help in getting a better culture swab because bacteria is not usually distributed evenly across a wound. Moving on to talk about the spectrofluorometer. A spectrofluorometer is similar to a spectrometer, which is what is used in labs mostly to measure absorbances. Even though a spectrofluorometer can also measure absorbances, it is also able to measure excitation and emission wavelengths inside the visual spectrum, and with limitations that vary between different spectrofluorometers, they can also scan into the infrared and UV spectrum to some extent. For my experiment, we scanned a special 96 well plate that has a clear bottom/top and black sides to prevent the wavelengths from bouncing off the sides. The spec would be hooked up to a computer, and different settings on the computer can be adjusted to get what you want to measure. The process of what happens inside a spec starts with a white light. In the example of measuring the fluorescent properties of bacteria, you would set the excitation wavelength to 405 nanometers, and the violet light would leave the exit slit and hit the sample. Then the porphyrins in the bacteria would emit a red light, and the red light will be diffracted and come out through the second exit slit. Then the detector would detect the red light being emitted at around 630 nanometers, and so there would be a peak in fluorescence intensity at around 630 nanometers. This is how a graph from a spectrophotometer would typically look like, with the fluorescence intensity on the y-axis and the emission wavelength on the x-axis. So using the concept of the fluorescent property of porphyrin in bacteria, there might be a fluorescent property in the biofilms as well. Some preliminary data that was done shows scans of a polymicrobial planktonic solution compared to a polymicrobial biofilm of S. aureus, E. coli, and E. cloacae. These scans were set to an excitation wavelength of 405 nanometers and an emission wavelength range spanning the visual spectrum from 435 to 695 nanometers. This setting that was scanned is the default excitation and emission that is used on the MolecuLight device in order to diagnose bacterial infections. One thing to note is that the fluorescence intensity of the biofilm graph is flatter than the planktonic solution graph. This region is considered noise, but it is something worth mentioning because the biofilm could be quenching some sort of signal in the shorter wavelength region, which could further be analyzed scanning at different excitation wavelengths. And just to prove that the bacterial emission wavelength is at around 630 nanometers. We can see a huge peak at around 630 nanometers on the two graphs. Since we don't know any components of a biofilm that can fluoresce like porphyrin, a future experiment would consist of different sets of excitation wavelengths along with the standard 405 excitation wavelength and different ranges of emission wavelengths will be measured depending on the limitations of the spectrophotometer. Our next step is to perform an experiment with 4 different conditions with the wound pathogens S. aureus, E. coli, E. cloacae, and P. mirabilis. The 4 conditions consist of monomicrobial planktonic solution, monomicrobial biofilms, polymicrobial planktonic solution, and polymicrobial biofilms. This field of research is extremely important because it would be the first real-time diagnostic test for biofilms, meaning that you can know instantly if someone's infection has turned into a biofilm or not, instead of waiting days for results and allowing the wound to get worse. Discovering a specific excitation and emission wavelength characteristic to biofilms could allow Moleculite to incorporate a biofilm mode on their device in order to diagnose a biofilm infection with the capture of one image. Because of a fast diagnosis, more lives can be saved by catching bad infections earlier before they get out of control and it's too late. Thank you guys for taking the time to listen to my presentation, and I am open to answering any questions that you guys might have. Thank you.